pleasure being here. Also excited maybe finding some synergisms or some collaboration between my research, which involves plant-associated bacteria, and your research, which involves more marine life. So as Giovanna said, I work in ICGB, which stands for International Center for Genetic Engineering and Biotechnology. This is an institution which was created in 1987 as a UNIDO special project. UNIDO stands for United Nations Industrial Development Organization. The reason for creating ICGB is because with the advent of recombinant DNA technology, the United Nations was concerned that this new technology, genetic engineering and biotechnology, which was emerging, that the developing countries would not benefit from this new, obviously providing new economic advantages. So in order to promote biotechnology in developing countries, we were created, organization. And we remained a special project of the UNIDO until 1995. And from 1995 onwards, thanks to our founder, Professor Arturo Falaschi, who ensured that we have a core funding from the Ministry of Foreign Affairs in Italy. We now are an independent international intergovernmental organization. We are actually, our organization is, as Giovanna said, our institution, I work in Trieste, okay, here, which is an institution of approximately 250 people, and is actually at the moment the headquarters of ICGB. So we have the main administration, but in fact our organization comprises of 3 institutes, one in New Delhi, which was also created at the same time of Trieste in 1987. At the moment is about twice the size of Trieste. You know, India has grown a lot in terms of biotechnology. It's a very powerful biotechnology country right now. So their funding, the funding is going very well in India at the moment. And since 2007, we created also a component in South Africa. Because in our view, some developing countries have made incredible progress in terms of biotechnology, and they're located in South America and Asia, but the African continent, in our view, is still lagging behind. So to give it some momentum, a new institute was created in South Africa, which is a little small right now, but we hope to bring it to the size of Trieste in the near future. So our organization is 3 institutes and 64 member countries at the moment. So in order to be part of ICGB, you have to sign an agreement with us, a statute, and we have, unfortunately the slides are not very, probably the incompatibility with Macintosh, but they're not very clear sometimes, but we have the green countries here are all countries which are member of the ICGB. So a country to become a member, they have to sign a statute and they have to sign an agreement with us. And you can see here, we really cover a lot of the developing world. And if you're a member country, you can benefit a lot. So what do we do in order to promote biotechnology in developing countries? So as I mentioned already, we have advanced research projects in Trieste, New Delhi, and Cape Town. So a lot of these projects, all these 3 institutes have PIs like me, and we run projects which are directed towards the research needs of developing countries as well as training. So I do a lot of training, a lot of young scientists come to our labs. So in fact, we have our own fellowships program for postdocs, and we have PhDs. And we have these fellowships for postdocs and PhD are only available for scientists of member countries. Okay? Of course, member countries pay a small fee to be part of ICGB, but I want to make clear that they get a lot more back than what they put in. Most of the funding comes from Italy and India and South Africa. We also organize meetings, courses, and workshops, about 25 a year, and we organize 5 or 6 in Trieste, but we sponsor about 20 workshop courses or conferences in our member countries. Okay? So also Italy is a member country. For example, if you want to organize a workshop here and you want to have some sponsorship from ICGB, you can apply for funds in our institute. We give up to 20,000 euros for this. I'm organizing a course at the end of this, at the end of November. This year in plant microbiomes. I'm very proud of this. We have our own granting system. So we have 1 call a year, and we give grants, research grants, 3-year grants to scientists from our member countries. It's very competitive. You know, we get more 400 applications, and we fund about 25 a year. Okay? And also we have other services. So you can see here we have multiple instruments of action to promote science in developing countries. Okay? So this is our website, and if some of you are also from one of the countries that are part of ICGB, and my recommendation is always to look at our website, because we, apart from our fellowships, you can maybe go to a meeting or a course of ICGB, and if you're a member country, we can sponsor you to go there to this. You can or apply for a grant or do other things. So this is all I want to say about ICGB, and I'm very proud because I think we are rather a unique institution. So going to the work I do, as Giovanna said, I'm very interested in signaling. I've been involved in this research for a few years now, and now I tell myself, very often during my research that bacteria are social organisms, and they like to live as part of communities. They don't like to live on their own. On their own, bacteria, in my view, are much more vulnerable to predators and are much less competitive. Whereas when they form a group, they can survive better biotic and abiotic stresses, and they can also in a much better position to attack a host or other communities of other microorganisms. And is now a lot of work has been done on in this field, and a lot of scientists have shown that bacteria produce small signals, and these signals accumulate with the bacterial community, and then there is a synchronization of behavior in response to this signal, to the concentration and the type of signal. And a lot of phenotypes have been associated with this behavior, like, as you can see here, production of a biofilm, production of a secondary metabolite, virulence, competition, symbiosis, all phenotypes which makes perfect sense to do when there's a community rather than when you are solitary. Okay, and a lot of again, my apologies for the poor quality of the slides. They're not projected very well, but I think we can see. But what I want to say here is that some of these quorum sensing signals have been elucidated, the structures, but there's a lot more to do. This is the tip of the iceberg. We're beginning to find more and more types of signals produced by bacteria. And for example, here are small peptides produced by gram-positive. They tend to be usually simple, and I work a lot with these kind of signals. They're homoserine lactones signals. They're rather simple. They have an homoserine, and they have then an acyl chain which varies in length, and at position 3 on the acyl chain there is a ketone or hydroxy, different substitutions. And these are very common among protobacteria. The only produced actually by protobacteria, gram-negative bacteria, which is the protobacteria class, and they're very actually simple to detect. You can see another picture here. Here is the first homoserine lactone signal, which has been elucidated from produced by marine bacteria, actually, Vibrio fischeri, symbiont of some squids. You can see this is conserved among all homoserine lactones, and this is a carbon-6 acyl chain with a ketone, so it's a 3-oxo-C6 homoserine lactone. But a lot of homoserine lactones have been found produced by a lot of species, bacterial species, and what varies is the length of the acyl chain. You can have up to C20, in fact. And at position 3, in this case, is a ketone. You can have an hydroxy or a methylene. So you can have, for example, 3-oxo-C8, 3-oxo-C10, or you can have 3-hydroxy-C6, or you can just have C6. So there's a lot of different dialects to this language, and lots of different homoserine lactones have been elucidated. So this provides some specificity, obviously. So you can see here, for example, the system. What I want to stress is the simplicity and the power of the system. So you have 2 genes, very, very simple, very powerful. 2 genes. You have a gene which belongs to a Luxi family, because Luxi is the first gene that's been discovered in Vibrio fischeri. This is a small enzyme that produces the homoserine lactone. I don't have time, but there's a lot of work about the synthesis. It uses 2 substrates which are present actually in every living cell, and this substrate is involved in either fatty acid biosynthesis, or this is S-adenosylmethionine, which is involved in a lot of enzyme reactions. So it takes these 2 opportunistically and makes the homoserine lactone. Low cell density, low number of cells, low concentration of the signal. My apologies again, is not very clear, but at high cell density you have a high concentration, and very important, this particular signal here, not all quorum sensing signal, but this particular signal can diffuse freely. Gram-negative have 2 membranes, outer and inner membrane. This particular signal does not need any active transport. high concentration of cells, high concentration of signal. The signal can diffuse in and out. It has a regulator here. This is should be a green circle, which interacts directly with the signal. This is actually in most cases covalent, very strong, irreversible. And this interaction is a one-to-one stoichiometry, makes a homodimer, and this guy can then bind DNA and switch on transcriptional genes, which are regulated by quorum sense. So extremely, extremely simple, extremely powerful. Two-gene system, one gene making the signal, one gene coding for the enzyme making the signal, another gene coding the regulator that interacts with the signal at high cell density and regulates gene expression. Here's another slide. You can see this kind of quorum sensing via homoserine lactone has been discovered in a lot of important bacteria. You can see here. And these are bad guys, except the first one, which is, I put it there because it's one that had started the whole research, this Vibrio fischeri. But you can see these are bad guys, and these are bad plant guys here. Very important plant pathogens. Also, I will not mention here the other types of quorum sensing systems that are used. And there are also very other important plant pathogens like Pseudomonas syringae, or Xylella fastidiosa, or Xanthomonas that I will not talk about here. So there's a lot of exciting research in quorum sensing in plant pathogens with very nice potential applications. But keep in mind that this kind of signaling with homoserine lactones, but in general quorum sensing, is used by a lot of good guys. We know that most bacteria that are associated with plants are beneficial to the plant, and in a way, unfortunately, these guys have a similar mechanism of communication. So this also obviously has implications in terms of applications, because we have to be careful. If we want to control a pathogen by interrupting or interfering with signaling, we have to be careful not to affect the beneficial community of the plant. Okay, so my work has been, I focus on rice, for obvious reasons. Rice is by far the most important cereal crop in the world. And when I look at the statistics, what our challenge ahead in the next 30, 40 years is scary. The amount of rice we have to produce with less land, more people. So look, having a more sustainable production and a higher production of rice is going to be fundamental for our health of the planet and of population. So I look at bacteria associated with rice, and when I started this work, I did some literature work, and I saw that there's some bad guys and there's a lot of good guys that obviously live with rice. It's okay? Okay. Okay. So we did a lot of work. My students did a lot of work, the people in my lab, on some pathogens like Burkholderias, Xanthomonas, and Pseudomonas, and this I'll be talking about later on. And also some nice guys, rhizosphere bacteria. Rhizosphere is the soil closest to the root. This is like, as I was telling the students earlier, there's a parallelism between the rhizosphere and our human gut. So plants exude a lot of components via the roots in order to recruit a healthy microbiome, a healthy microbiota. And there's a lot of good guys that we have to study. And also, I'm very interested in endophytes. Through the roots, lateral roots, some bacteria can get inside the plant, live in intercellular spaces, and in some cases even move upwards and colonize all the plant. These endophytes, bacteria get in. A lot of these are good guys, and maybe we can use them as bug probiotics. And I'll be talking very briefly at the end, 3 or 4 slides, about a project that we have in our lab, an applied project on this. So we started working on this, and I don't want to now just spend time on just giving an account of what we've done, but what we've done here is we took a lot of these bacteria. I spent a lot of time getting these strains from all over the world. All these pathogens are beneficial from colleagues, and then we see, oh, they have signaling, they produce a signal, we knock out the genes, then we do plant studies and we see the effect, but all on a very, what's it called, in my view, unrealistic scenario, right? So I want to discuss now what I'm actually looking at now in thinking more of what happens in the wild. Before I do that, I want to just mention a couple of slides looking at quorum sensing in these guys. We embarked upon something new, something we didn't expect, which I just want to mention here quickly with 2 slides. And this is the beneficial pseudomonads, rhizosphere pseudomonads, and the xanthomonas oryzae, which is a vascular pathogen of rice. This is the second most important pathogen of rice after a fungus. This is a serious guy. This creates 10% loss in Asia, China and Vietnam and India. It's a very serious pathogen. It goes through the hydatodes in the leaves, enters the vascular system and blocks it. We didn't find in these strains, belonging to these species, Pseudomonas putida, or anyway, the general Pseudomonas and the Xanthomonas oryzae, we didn't find the ability to produce AHLs. And when we started this work, we didn't have much genomes, basically. Now we do. But then, I was mentioned my student Sarah. She spent a year looking at 25 strains that we got from Bayer, crop science, very pathogenic, isolated in India, and lots of strains from rice rhizosphere of these Pseudomonas, and we couldn't detect any homocyan lactones. And that's very frustrating because when you have a negative result, it could mean, oh, maybe they're making a new homocyan lactone. But then the genomes came out, and what we saw in these genomes was something that really baffled us, is that they didn't have the ability to make the signal, but if you remember, there are two components, making the signal and detecting the signal. But what they had in the chromosome is a gene that coded for a regulator that had an homocyan lactone binding domain. This is a typical structure. This is the N terminus, this is the C terminus of the protein. And it's a modular protein, these proteins that bind the homocyan lactones. And they have an homocyan lactone binding domain and a DNA binding domain. And they had in their genome a gene that perfectly matched this scenario. And it was basically a guess that, wow, there's a protein that can respond to the signal. So basically, I can listen. I don't speak, but I can listen. So we were baffled, and we thought, wow, it must. What's happening here? A response to AHLs made by other bacteria, which makes sense in interspecies. And this is also thanks to a good friend of mine, Brian Hammer in Ohio State in Columbus. He works with E. coli. E. coli does not make AHLs, but can respond to them. It has a protein, a regulator that can detect them. And he has shown that E. coli detects signals, homocyan lactones from its neighbors. So it lives in an interspecies community, and it responds to signals made, AHL signals made by other neighboring bacteria. So we thought it might be similar. Or, I put it here, but this is a cheat because I put it afterwards. We thought first this was a scenario, or it responds something from the plant, because there were reports, by Max Tepliski in the States that plants maybe make homocyan lactones. So to make a very long story short, to make 2 PhDs and 2 postdocs, so one slide is that we worked with yeah, this is sad, isn't it? I mean, it's a lot of, we showed that the beneficial guy, Rhizosphere pseudomonas, and the Xanthomonas oryzae pathogen, they have a Luxar regulator, which evolved away from binding homocyan lactones, and now they bind a plant compound, and by doing so, they regulate virulence, or they regulate beneficial aspects. In this case, they regulate the production of secondary metabolites, which are antifungal compounds. So this was really exciting, and we got a lot of interest from the scientific community. People were so excited. I got invited to a lot of places. It was exciting. In fact, we published a novel, Widespread Interkingdom Singling Circuit, and again, I apologize here. But what was really exciting, then when we went to do bioinformatics, we found that this kind of protein, which had some little differences in amino acids in the homocyan lactone binding domain, because now it's binding something else. We found it in a lot of bacteria, but you cannot see here all these bacteria, Xanthomonas, Pseudomonas, Rhizobium, Bradyrhizobium. They are all plant-associated bacteria. So we stumbled across a new system of interkingdom signaling, so detecting a plant compound rather than a homocyan lactone. And I'm collaborating with P. Greenberg. We think this is a peptide, and in fact, the last issue of PNAS, they publish a signal that this system responds to. Okay. So close the bracket. Now I go back to what I want to discuss now is where I'm going now, and where I'm going now is really that I want to try, and I'm finding it very, very, very hard. I'm trying to look at a more realistic scenario. A lot of microbiome studies are coming out, and we're seeing that plant-associated bacteria live in complex multi-species communities. So what's happening? How are these communities communicating? How are they interacting? And keep in mind that these plant-associated communities have evolved together for millions and millions of years next to the plant. So the plant will just not sit there and take all this. probably it interferes with the signaling, or the signaling molecules have an effect on the plant. So there's a lot of studies now coming out that scientists are looking at the bacterial signaling molecules, what effect they have on the plant, and they do have an effect. What does the plant do to these signaling molecules? Do they produce similar? So there's a lot of work to be done here, because there's a complex interaction between the bacteria and the cell, between the different species, and with the plant. This is the direction I want to take. So I'm gonna talk for the next 20 minutes about something we're doing and we intend to do. The first story I want to tell you is that, again, by chance, I embarked on what I think is a really nice story. Is that I got an email a few years back by Taha Hosni at the University of Perugia, who was studying this disease called olive knot, rogna dell'ulivo in Italian, the olive knot disease. So basically, this is a Pseudomonas savastani pathogen, Pathos savastani, that lives on the surface of twigs. And when there is a damage by weather or by even humans, there's an opening, and it can get inside. It causes apoplasia by it colonizes, produces plant hormones, causes apoplasia, and it causes these olive knots. It's actually not a serious disease. If you're a good farmer, you just cut it off in a sterile way and burn it, forget about it. If you're not a good farmer and forget about it, this can spread, and you can see here a tree that is widely infected, and then if you leave it bad enough, it even affects the olive production and so on and so forth. So what Taha said, he's a student 2 meters tall from Morocco. He said, can I come to your lab, because I have isolates of this guy and I want to see if they produce homoserine lactose. And we have all this, we have all the setup to detect these molecules and to make mutants. So he came to my lab. He stayed actually 6 months, and he was extremely productive. So the first thing we found that Pseudomonas savastani makes C8 3-oxo, I'll show you later, homoserine lactose. It makes a signal. So then we made the mutant. We inactivated the gene in the chromosome that produces the signal, and we inactivated the gene, the regulator that responds to the signal. So we have basically a signal negative mutant and a signal blind mutant. So, and then we sent these mutants back to Perugia, and in the group of Professor Bonaiuro, and he infected, these are 1-year olive plants, he infected the plants with the wild type, and we wait 60 days, not 60 hours, and you have an olive knot being formed, and you can measure the size of the olive knot. Then you infect with the signal negative or the signal blind, and look, the knot is much less. So great, we have hormone sensing, we make homoserine lactones, and this communication is important for the bacteria to infect and to develop the knot. Okay, this was, again, this is great, but it was not revolutionary, not very recent. There's so many pathogens that have been shown that have homoserine lactones, and that their homoserine lactone, the signaling is important for pathogenesis. Pantoes tuarti, Erwinia carotovora, Agrobacterium tumefaciens, Pseudomonas syringae. So this was just another one. And I told Taha, I'm not interested in what gene it regulates. It might regulate some hormones. I'm more interested in communication interspecies. And what he told me, which got me really excited, is that what he did in his PhD, he went to the wild, and he isolated bacteria from the olive knot. And of course he isolated the pathogen. But he tells me, I also isolate very often under another bacterium, which is called Erwinia toletana. And then we went to the literature, and we saw actually Erwinia toletana, after the city of Toledo in Spain, was classified as a novel species in 2004 as a toletana novel species associated with Pseudomonas savastani-induced tree knots. And this Erwinia toletana is both an epiphyte and an endophyte of olive trees. And it's absolutely harmless. It's beneficial. There's reports in the literature on this. So I begin to think, wait a minute, what's happening here? Bacteria are not stupid. So here we have an olive knot, which is something that the pathogen makes with a lot of energy. And inside the olive knot, there is an Erwinia toletana. Trust me, there's a lot of social microbiologists that study all the time. Bacteria are very smart. They don't work for others. They are now in place beautiful mechanisms to ensure that they're public goods. So what they produce for the outside, their public goods are for themselves as much as possible. They don't want cheats to come and take their public goods. So I'm saying, and then Taha is telling me, oh, but I found a lot of Erwinia. So what is this guy here? Is there any signal between these two? Is this a friend or is this an enemy? Is this a cheat or is this a cooperator? So we started working on this. And the first thing Taha showed is that great. The Erwinia toledana has quorum sensing. And guess what? It makes exactly the same signal of the Pseudomonas salustianum pathogen, a C8-3-oxo, which could be by chance, but that alarmed us. So then, just like in the pathogen, with this Erwinia, we made the mutant that doesn't produce a signal negative, and then we made the mutant that doesn't make the regular signal blind. And what we started to do together with Roberto in Perugia, we started to do co-infections. So we infected the one-year olive plants with the pathogen together with the Erwinia. And the first thing we see here, if we use the two wild types, we have a much bigger knob. Well, bigger, I mean, in plant pathology terms, this is a significantly bigger knob. So that was great. We have a co-infection, so we give a lot of both, the knob is bigger. But then what really got us really excited, and I already showed you, if we